The clinical drug interaction dataset in the DIDB contains results from in vivo drug interaction studies where a drug is tested as a perpetrator or victim and results are organized according to the overall effect and the underlying mechanisms of the interaction. This video is going to show you how to access the information extracted from a clinical DDI from a single source and how to compare data for one or multiple compounds from a large set of different studies. For this tutorial, I chose the drug idelalisib, a kinase inhibitor approved in 2014. First, we can use the main search function to find a specific article, for example by PubMed ID number. But it's more likely that we want to look at all of the available information for a specific object drug. The easiest way is to go to All Queries, and under Basic Queries, select Drug Queries. Let's look at the information retrieved when idelalisib is tested as a precipitant. Type in the drug name and select from the drop-down menu. Then select only in vivo studies and click Submit. The query results are presented by default in a table. From the first column, you can see that the studies are organized by overall effect, positive or negative, based on the 20% cutoff change in exposure as measured by the AUC or clearance, and according to the mechanism underlying the overall effect. You also have the option to view the results in a tree view. In the table view result, the column accession number, or NDA BLA number, shows the source of the information. In the case of midazolam, 2 citations are listed, one coming from the NDA documents released from the FDA, and the other from the literature. Each citation number is a hyperlink that allows you to access the citation details, the PK profile of the citation drugs, the PubMed or FDA portal from which the information comes from, and Semantic Scholar for literature studies. Let's review the citation details coming from the PubMed ID number. On top of the new page is the full reference of the article that is directly extracted from PubMed. Below is all the information in tree view that is manually extracted from the article. In this citation, idelalisib was tested as a precipitant with 3 different objects presented in alphabetical order. For clinical studies, the first few lines describe the study methods, including study design, population description, and how the object and precipitant drugs were administered. For each study, results are first presented in the control state, in our case when midazolam is administered alone, and then during the test state when midazolam is co-administered with idelalisib. Highlighted in green is the percent change in exposure directly derived from the geometric mean ratio test over control phase, or calculated as change in AUC, clearance, and plasma concentrations by the DIDB team, and allows for comparison between studies across multiple citations. When information for relevant metabolites is available, it is also presented. Lastly, when investigated and reported, the effect of the DDI on the pharmacodynamics of the object and a description of side effects are also displayed. In our example, based on change in AUC, idelalisib appears to be a strong CYP3A inhibitor. Now I'm going to show you how to retrieve other strong CYP3A inhibitors on midazolam. To retrieve the specific list of inhibitors, I am going to use the AUC clearance change query that is directly accessible here. First, click on the appropriate tabs. With Objects, find all studies providing the percent change in AUC, enter the drug, midazolam, with inhibitors, where the value is greater than or equal to 400%, which corresponds to a 5-fold increase, and click Submit. In the Results tab, the results are presented by default in a table, starting with the overall effect in the first column, followed by the object, in our case midazolam, and so on. You can also review the results in the tree view. However, the table view feature allows for an easier comparison between inhibitors using the percent change in AUC. You can use the table features to customize the presentation of the results to your liking. You can move columns, reorder data, or filter. Once you are satisfied with the layout of the table you have generated, you can copy it or download it as an Excel or a CSV file, or copy the link and send it to a colleague. And that concludes this video tutorial on the clinical drug interaction dataset. I invite you to check out our other video tutorials, which provide an in-depth look into the content and functionalities of the DIDB. If you have any questions, just go to our website, druginteractionsolutions.org, and click on Contact Us.